Welcome back to this last video in the third section about energy. And in this, we're going to look to see what happens in the cell after glycolysis when there is no oxygen. So if you remember this slide, what I said previously was after glycolysis, oxygen makes a difference because in the presence of oxygen, the pyruvate was going to move into the cell and we would have the linking reaction followed by the Krebs cycle and the electron transport chain. But if we don't have oxygen, we are going, instead of having those three steps, we're going to have a process known as fermentation. So in fermentation, all we have is the energy harvested from the sugar without oxygen from glycolysis. And then we're going to take that pyruvate and do something else with it. And we have two options. Option number one, we take that pyruvate and we take 2 carbons off of it. So in other words, we're going to make 2 more carbon dioxide. So we can make bubbles if you want and convert it to ethanol. Now remember in glycolysis, we had made NADH. Previously, that NADH can be returned back to glucose. And so it can just be recycled. So with fermentation, that is the first option, create alcohol. The second option without oxygen is to take that pyruvate and to change the molecule slightly to make lactic acid, also known as lactate. And once again, the NADH from glycolysis is not used. It is just returned back to become an electron acceptor once again. So when the cell doesn't have oxygen, there's only 2 AT produced for every glucose. There's no new ATP. The only ATP were the 2 produced from the process of glycolysis. So the net results of fermentation were lactic acid fermentation in your muscle cells. Have you ever had those cramps? That means you need to stop and breathe. You need to get some oxygen going to those muscles. I know everybody says you need to drink. No, you need to breathe. Yes, let your circulation go around and get some oxygen going to your muscles. With bacteria, for instance, Aspergillus, with the absence of oxygen, we can make some soy sauce. If we did lactobacillus instead, we could make cheese or we could make yogurt. If we use yeast, we could make beer or wine or bread. Have any of you ever tried to make, or any of your parents or aunts or uncles try to make alcohol like in your bathtub? Oftentimes, when people first try to make it, it doesn't work and they think there's something wrong with the kit. Well, the reason why it's not working is because air gets into it. Remember, fermentation only occurs in the absence of oxygen. Okay. So you get alcohol with fermentation by yeast. Lactic acid is when fermentation occurs in your muscle cells or with bacteria. So take a moment and review the differences between cellular respiration or fermentation, where it occurs in the cell, and the net ATP usage. And so let me kind of pull this all together and go through this review with you. If I put these here, I want you to take a moment and go over it. So I'm going to go over this one with you just because this video is so short. With aerobic cellular respiration, it occurs in 3 main phases. The first phase was glycolysis, where we start with a 6-carbon sugar of glucose, end up with 2 3-carbon pyruvates. This happens because glucose splits, and when it does that, in addition to ending up with a 3-carbon pyruvate, we're going to end up with a net of 2 ATP as well as NADH, which are electron carriers. The pyruvate is going to be oxidized. It's going to yield us a carbon dioxide, as well as giving us an acetyl-CoA, which enters the linking reaction, which basically occurred during the linking reaction. Acetyl-CoA then enters the second main stage, the Krebs cycle, also known as the citric acid cycle. And remember, we have two pyruvates, so citric acid cycle happens twice. The end of this, we would end up with two new ATP. We'll also end up with more carbon dioxide, more NADH, and more FADH2. So the NADH and the FADH2 from both glycolysis and Krebs cycle will enter the electron transport chain. Now remember, the electron transport chain is going to be going through twice at least, because the Krebs cycle goes through twice because of the pyruvate. At the end of the electron transport chain, we are going to be generating from hydrogen coming off the NADH and the FADH2 a proton gradient, which eventually comes back across and is used to generate new ATP. The electrons that were donated are going to be used to bind to oxygen, generating our waste product of water. And remember, FADH2 with the little 2 after the H only makes 2 ATP, whereas NADH will make 3 ATP. And with that, we are done with everything we want to learn about energy. I thank you for all your hard work, and we will come back, and our whole next series of videos will be on the cell.